Hello everyone, good evening and welcome to 9th and 10th channel of BYJU'S. I am the teacher Ankita and I welcome you all in today's class. In today's class, we will be discussing about all these cells organelle. It's a very important session everyone, so please make sure to stay with us till the end. If you're new here, do take a moment and hit that subscribe button and don't forget to hit the like button for the video. Good evening everyone, good evening. Quickly ask your friends to join the class, right? 6 p.m. is class, 9 day, and everyone, I'm meeting you, like especially for biology, after a long time. Yeah, we have been meeting very regularly for SST. Sometimes I feel that, yeah, we don't meet often for bio classes. Yes? How are you all? Good evening, everyone. Good evening. So today's class is very important and it's a very, fruitful class, I would say. We will be focusing on all the cell organelles, right? A very quick recap of all the cell organelles and some bonus information also. So for that, you have to stay with us till the end. Good evening, everyone. Good evening. Hello. How are you? Awesome. I can see that so many of you have joined, everyone. Please make sure to hit the like button. We should at least have 100 likes. By the end of the session, that's very small ask, right, from your teacher. So everyone, please make sure you hit the like button. I'm good. Thank you for asking. Yes, I will be replying, everyone. Don't worry. Yes. I'm good, everyone. I'm good. Good to see that all of you are excited. I hope that all of you are excited for this class, right? And I hope that all of you can see me and can hear me clearly. Definitely, we have been talking, so I know that you can. But yeah, I hope that you can see this cute little smiley. Awesome everyone. So now that we are there, right, let's quickly move ahead and I'll not be taking much of your time. So everyone, we know that, right, you know that this is our channel 9th and 10th and we have classes from 6 to 8 p.m. right? And of course 5 p.m. we have shorts and sometime we do take extra classes. So from 5 p.m. everyone, we have active classes, right? And of course, yeah, and it's a good time for all of us to be here. And of course on this channel, we make sure to give you the quality content, right, and that actually help you to excel in your academics and of course in your personal life. I hope that all of you see this timetable, right? Take a screenshot, everyone. If you're new here, do take a screenshot. Keep it with you. We follow this timetable, right? So here we go. So everyone, are you ready? Now I'm starting, right? I want all of you to stay focused. It's a 1 hour long class, right? It's a 1 hour long class. We will try to finish it before 1 hour. I need your help for that, right? Topics are really very easy. It's a very quick recap. So everyone, we will be starting. Class will be of 1 hour. Yes, and people are asking, ma'am, Riddhi is asking, ma'am, you like bio or SST? I like both. Yes, good. Nishant, we will be starting class 10th ka second part also, don't worry. Okay, good to go. Yes, I can see that. Thank you. I'm sorry, I forgot. Yes, awesome everyone. Chaliye, let's get started. So I will be taking your doubts, but after a while I will tell that it's a doubt. I will be asking you for the doubts and then only please write your doubts, okay? But before that, don't say, ma'am, you're ignoring me. See, I have no intention of doing that, okay? So let's get started. So everyone, we started this chapter with the cell, right? We know that cells are the structure and the fundamental unit of life. We know the history of the cells, right? And we know that there are very important parts. We have discussed about the nucleus, we discussed about the cell membrane, and of course, the other things, right? Like the cytoplasm is, I'm sorry, this is incorrect. It's supposed not to be the mitochondria, right? It is the cytoplasm. So we have discussed all of these. Now what we'll be doing in today's class is discussing the cells organelles, right? Now why we call them as cell organelle, everyone? Do you have any idea? Right? We call them as cell organelles. So in our body, everyone, we have different parts. We have stomach, we have liver, we have heart, we have lungs, and all of these are the different organs that are present in our body that actually help in performing some functions, right? Similarly, in the cell, right? Similarly, in the cell, we cannot call them as the organ, but they are the organelles, or we can say there are small parts inside the cell that actually help them perform certain functions. So, the cells organelle, of course, in the cells we will be discussing about it. So everyone, I hope that all of you are clear. Yes? Yes. Very good, very good. Good to see that all of you are answering over here. Very good, everyone. Now, let's take the first First cell organelle, everyone. Okay, do you want to pick your favorite cell organelle or we'll just start randomly? Right, everyone? After the nucleus, which organelle usually comes? And whenever we talk about the organelles, right, there's a one important part that we always start with. I can see some name. I can see ER, mitochondria. Okay, mitochondria and ER there. They are the winner now. Very good, very good. Okay, I think ER is winning and it's mitochondria. ER, mitochondria. Nucleus we're not considering. Nucleus over here is definitely a very important part, but we don't study it under the organelles. Everyone is saying mitochondria. I have ER over here, so let's study ER. ER is also winning. So here, everyone, let's talk about the ER, the endoplasmic reticulum. Now, I'm sure you are aware about the ER. The short form is ER. Right? Endoplasmic reticulum. Yes, we'll have the mitochondria. Don't worry, we'll have the mitochondria also. So everyone, when we talk about the structure, right, this is how the ER looks. Most important thing about the ER is that it has a membrane found. So can you see? Membrane bound tubes and sheet. So here we can see that membranes are there. They are either present in a tube form or in the sheet form. Yes? Okay, awesome. Got your point. So of course, see over here we have the sheet, and over here we can see the tubes. So of course, it's a structure which consists both of them. Like a sheet is also there and the tube is also there, right? And then of course it appears like a long tubule. So yeah, we can say that it appears like that. Then of course, in that also we have two things, right? We have smooth ER and we have rough ER. Okie dokie, everyone? Yes, right? We have rough ER and we have smooth ER. Rough ER is called as the rough endoplasmic reticulum and smooth ER is called as smooth endoplasmic reticulum. It actually helps in the synthesis of the manufacturing of the proteins. Rough ER and smooth ER help in the formation of the lipids. Very good, everyone. Very good. So this is very easy. I know that. Very good. You all are giving the answer. Okay, everyone. So we have just discussed the function. We have discussed the function of the ER in general. Now, apart from that, can you tell me, can you help me that what is another function of the ER? Yes, 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 very good. Everyone, I can see your answers. Very good, everyone. Very good, very good. Membrane biogenesis, very good. Cytoplasmic structural framework, awesome. Very good, I am very proud. Okay, very good, everyone. So let's see the other important function of the ER when we discuss about apart from the manufacturing of proteins, right, and lipids, is that they play the very important role, right, in the structural framework. So the ER help in providing the structure. Now, can you see this particular structure? They're very closely connected to the nucleus, right, and this is a very prominent structure. So it provides a skeleton framework. Very, very important. Okay, I can see all of your answering. I will take up that doubt. One doubt which is very important before we move ahead is what are lipids? Everyone, what are lipids? We usually confuse them with the fats. So we can, if we have to say, right, we can say that they are the fatty. They have the oil compounds, right? They are not soluble. They are insoluble in water. Okay? Yes, fatty substance we say. Fatty substance like oil, we have wax, that we have as the lipids. Compounds in the fatty acid, absolutely correct. See, I think if you have doubt, I would request you to watch the session that we did on the cell membrane. There we have gone detail and understood what are the lipids. Very good. Yes, Rajiv, we have just discussed. We have just discussed what are the lipids. Okay. So now everyone, we are clear that what are the lipids? Good evening. Yes, welcome to the class. Okay, very interesting question. Okay, yes. Now let's take a look, everyone, over here. Now we'll be focusing on the structure now. Can you see the structure? Can you see the structure of the ER, everyone? Yes, yes, very good. Now tell me, everyone, how many membrane you can see over here? Right, I'm sure you can see the membrane, but how many membrane you can see? Focus, if you look closely, what you see, everyone? There is one membrane which is outside. Let me just change the pen color. Give me a moment. Right, we can see that there's one which is outside and there's one inside. Yes or no? Yes or no? So how many membranes ER has? We have the answer. How many membranes ER has? 1 or 2? Double membrane. Very, very good, everyone. So the ER, right? When we talk about the ER, it is double membrane structure. Good. Very good, everyone. Awesome. So this is clear. Now let's take focus and let's quickly look at the functions of the rough endoplasmic reticulum. So of course, rough endoplasmic is called as rough. Can you tell me, everyone, why? I'm sure you have watched the sessions, right? Why we call it as the rough endoplasmic reticulum? I don't know who is saying infinity. Yeah, I know. Very good, because it has the ribosomes on it, right? These ribosomes are present on the rough endoplasmic reticulum, RER, and because of that we call them as the rough endoplasmic reticulum. They're not smooth, right? They're rough. Okay. Now let me ask you one more question, everyone. I'm just checking your knowledge. Where are the ribosomes produced? We have studied it, right? I'm sure you remember this. Where we'll see the synthesis or the formation of the ribosomes. Okay, I'll count till 5, everyone. 1, 2, 3, 4 and 5. I'm sure you have the answer now. The correct answer is nucleolus. Absolutely correct. So ribosomes are produced in the nucleolus, right? This is really very important for us to remember. We have studied that we have the nucleus, right? Inside the nucleus, what we have? We have the nucleolus, right? And inside this nucleolus, we'll see the synthesis of ribosomes, and these ribosomes will move out of the nucleus and will come and get attached on the rough ER, right? And that's how we see the formation of proteins. Everyone, are we clear with this? These ribosomes plays a very, very important role in the synthesis of proteins. Okay, so they are present on the ER. So let's make a flow chart, everyone. Let's make a flow chart. Right, so what we have a nucleolus, then we have ribosome on rough ER, and they make proteins. Everyone, are we clear? Quick thumbs up, we are clear with this, right? A small chotu sa flow chart so that we'll remember. Synthesized in the nucleolus, ribosomes will move out of the nucleus, get attached on the ER, then now they become rough endoplasmic reticulum, and they help in the synthesis of protein. Ta-da! Important function of ribosome. Now let's talk about the rough ER. The main function is that it actually helps in the protein synthesis. That's we all know. We all know, so we'll not be spending much time over here. Let's talk about the smooth ER. Now of course, why we call smooth ER? Because it has no ribosome. The surface is really very smooth. So for example, everyone, over here I have the duster, right? I hope that you can see this. This is a smooth surface. You know what? And this is a rough surface. See, rough surface, can you see? And this is a smooth surface. Of course, over here on the smooth ER we don't have the ribosomes, right? And this structure of course has more of the tubes. We can see, right? So can you see these tubular structures? And they're very smooth. They don't have the dots. These dots are nothing but the ribosomes. So that's why we call them as smooth ER. Now let's talk about the function of the smooth ER. Very, very important function, everyone. It actually helps in the synthesis of lipids. And everyone, we will see that it actually helps in the process of detoxification. Very good. I can see, I love you all. Hearts, everyone. Please send purple hearts from my side to you in the chat, because I can see you're giving all the correct answers. I'm very proud of you. What is detoxification? Basically clearing out the toxin from the body. They are actually present in the liver cells. Of course, all the cells have smooth ER, but in liver cells, there's huge amount, right? The number of the smooth ER, right, is definitely more, because they are the one that actually help in detoxification of the drugs. So, what happens? It actually break down the drugs, right? Make sure to clarify all the toxic from the body. So it's a very, very important function. We usually forget this function about the smooth ER, but this is super, super important. Everyone, thumbs up, right? Are we clear with this? Okay. So very quick recap. ER is very close to the nucleus. Two types, rough ER, smooth ER. Rough ER have ribosomes, help in the synthesis of proteins. Smooth ER, no ribosomes, help in the synthesis of lipids and in the detoxification process. Thank you everyone for the hearts. Now, if we combine lipids and protein, everyone, what we form? Can you tell me? We have lipids and we have protein. If they combine them, right, which structure we can get? Think about it. Liver cells. Plasma membrane, very good. So everyone, apart, right, apart from the formation of lipids and apart from the formation of the proteins, these two, right, the ER, smooth ER and rough ER performs a very important function for the production of proteins and lipids, which of course we know that are used in making the plasma membrane, right? And we call that whole process as the membrane biogenesis. Everyone, this is super important. Remember the word membrane. We know the membrane. Biogenesis, making of it, right? So it actually helps in the formation of membrane biogenesis, right? So what we have? We have the lipids. Over here we can see the lipids. And of course we have the proteins. Clear? So there are three important functions of the ER. First, synthesis of proteins. Second, synthesis of lipids and detoxification process. Third, membrane biogenesis. Clear? Clear as water. Yes, very good, very good. Ma'am, lipids are hydrophobic, right? So how come they are soluble in water? They're insoluble. But I said that they're insoluble. Right, if you see over here, right in the picture, we can see that they have the hydrophobic tail. Right? And over here we have the head. Clear? Yes, Riddhi, I am. Very good tech word. Okay, Janna, Jatin. Okay, unicorn, Arohi. Clear as water. Very good, Harsimran. Yes, we'll do that. Yes, we'll do that. Yes, Rajiv, Amisha. Okay, let's move ahead, everyone. So remember this. Take a screenshot of this. I'll move aside. Everyone take a screenshot. Very, very good. Okay. So we saw the synthesis of this, right? And of course, we see that there's a Golgi apparatus that actually help. The next organelle that we'll study actually help in the transportation of the lipids and the proteins. Now, the final function, everyone, it is provide the cytoplasmic framework, provide the surface for the various biochemical reactions also. Ta-da! No, they're not soluble, Ishika, they're not soluble. Okay, very good. Now, everyone, I think the next was on the mitochondria. So let's talk. Now, again, this is also super important and super easy. We know that, right? Mitochondria does what? There's only one function mitochondria have. Can you quickly tell me, everyone? Mitochondria is also called as the dash of the house. Yes? Mitochondria is called as the powerhouse of the cell. Absolutely correct, everyone. Now, this is a very, very amazing organelle, I would say. Very unique. Why? Because it has its own DNA. So, the structure of the mitochondria, when we talk about it, has two membrane, right? We can see that it has two membrane, inner membrane and the outer membrane. Inner and outer membrane. Apart from that, if you look closely, we can see that mitochondria, right, has these turns, these folds, and that we call it as cristae, right? These are present so that there's an increase in the surface area for the production of ATP. See, I'm sure if there is very, for example, if you have to, let's suppose, see, we have this thread. These are the actually headphones, not thread. See, we have, okay, this is, if you talk about, see, this, how, see, this, yeah, it's a long thread, right? But if I have to pack it into a small package, what will I do? I will fold it, right? I will try my best to fold it. Now, what will happen? Definitely when we are folding, we have more surface area, and in a case of mitochondria, these actually help in increasing the surface area so that there's more production of the ATP. Clear, everyone? Yes, everyone, are we clear? So there are two important words, right? Cristae. Remember that. Okay? They increase the surface area for the production of ATP. Then of course, you know what? These organelles are really, especially mitochondria, super interesting and super amazing. Why? Because they have their own DNA. Everyone, they have their own DNA, right? The mitochondria, and they actually help in making their own proteins. They don't have to wait. They don't have to wait for the nucleus and other organelles to send the material. They are sitting around saying, you know what they're saying? Yeah, we are self-capable of doing everything by ourselves. We don't have to wait. So, that's why right? They have their own DNA so that they can make their own proteins, ribosome, etc. Then of course when we talk about the structure, right, they definitely have fluid inside them, which is we usually call as matrix. Clear? Right, the fluidy, of course it has some fluid which is called as matrix. Clear? Good, very good. And of course it helps in the production of the ATP, adenosine triphosphate. Remember that everyone, I'm sure you all know that, right? ATP. Right? So all the energy that you and I required for the various reactions that are happening in our body comes from the mitochondria, and we call it as ATP, adenosine triphosphate. Remember this, okay? ATP, the energy currency of the cell. Very good. Okay. So now everyone, we have discussed about the two organelles, right? And I'll take a quick break over here. Not a break, a very quick time for us to discuss. Okay everyone, quickly tell me if you have any doubts. For mitochondria and ER only. Thank you Asa for helping your friends. We will be discussing that. Riddhi, a little bit. Okay. Define ER, we'll see that. Yes Shraddha, but they are present in the mitochondria and cistern are there in the Golgi apparatus. Yes. Mitochondria ATP, yes. Starfire, mitochondria help in the production of ATP. Riddhi, I've answered your question. DNA we have discussed in the previous class. Yes, mitochondria have its own DNA. How we can define rough ER? Of course, it's a type of the ER which have the ribosomes on that, which we call it as the rough endoplasmic reticulum, and they help in the manufacturing of proteins. Yes, Priya, they has their own DNA. Everyone, are we clear? Very good, sweetie. It is also called as the semi-autonomous organelle. Absolutely. Yes, they have their own proteins. Okay, very good everyone. SER function. SER is a smooth endoplasmic reticulum. Help in the manufacturing of lipids and help in clearing out the toxic from the body. Many of you are asking, ma'am, what is the name of the liver cell? You will be learning higher, but when we talk about the cells that are present in the liver are called as liver cell. Simple. Matrix is nothing but the fluid, right, which is present. Vaishnavi, I've answered your question. Saloni, matrix, I've answered your question. Yes, awesome. Fluid means fluid, not very solid, not very liquid. Like a jelly thing we usually say. Okay, I think all your doubts are there. I can see many of you have joined now, everyone. So you can go back and see. We will be having a very quick recap at the end, right? So I hope all of your doubts are clear. Ankit, I've answered your question. Yes, awesome, awesome everyone. Okay, now let's move ahead to the next organelle. I want all of your attention. Right, it's a quick break time everyone. How many likes we have? How many likes we have? What does ATP do? ATP actually help in the reactions, right? For example, when we eat food, we need to get the energy out of it. So the energy process, the energy production process happens, right? And we have the ATP, and without ATP, we cannot do anything. All the reactions that are happening inside our body need some action, need some energy. So we need ATP. It's an energy currency. Come on, everyone. How many likes? Okay, I'm sorry. I got distracted over here. How many likes, everyone? Wow, very, very proud of you, everyone. Let's have 100 likes. Full form of ATP, Adivika. It's over here. Adenosine triphosphate. Awesome. No, we can call it that same way. Golgi apparatus or Golgi bodies. That's a next organelle, everyone. So let's get started. Okay. Dextre, that is nothing but the folds that we have in mitochondria, bache. Okay, now we'll be discussing about the Golgi apparatus, everyone, or Golgi complex, or Golgi bodies also, right? So different names we have over here. So of course it was discovered by who? Yeah, we have the answer. Yes, we have the answer over here, everyone. So of course it was first described by the Campion Golgi. Remember the name Golgi, okay? Super important, very important, and very favorite organelle, I would say. Now everyone, when we talk about the structure, right, of the Golgi apparatus or the Golgi bodies, what we see, there is the, first of course there are the cisterns, right, cisterns. Now over here, can you see the cisterns? There are two words, everyone. In the, right, in the mitochondria, what types of fold we have? We call them as cristae. right? और यहाँ पे we have cistern. Everyone, I hope that it's clear. Clear, easy peasy, cistern. C, right? I-S-T-E-R-N-S, cisterns. Cisterns are present in where, everyone? Where we have the cisterns? Cisterns are present in the Golgi apparatus. And what are cisterns? Nothing but the structure. Can you see over here? If I stack like this, right? If I have, yes, these are parallel to each other. Yes, tell me, these are parallel to each other. They have membrane, right? These are the vesicles which are stacked onto each other. Now these are called as what? These are called as cistern. Everyone, are we clear? Clear? See, these are cistern, right? And what we have in mitochondria? The folds. Clear? Now it's very clear, right? So we can compare the structures also. So in mitochondria, it doesn't look like a mitochondria, I'm really sorry. We have these folds, right? What we call them as? Can anyone tell me what we call them as? Clear? And what we have in the Golgi apparatus? What we call them as? Cistern. Clear? See, you will remember this. Okay? Clear? This we have in the mitochondria, and this of course we have in the Golgi bodies. Good. Okay. Now that we are clear with this, everyone, let's see the structure. So of course we talked about there's a lumen, the small gap, right? These are stacked onto each other, there's a small gap. And very, very important thing that we have in the Golgi apparatus is that they have vesicles. So whatever they are making, right, they are forming the vesicles. For example, everyone, let's suppose, okay, how many of you, though it's not a very good habit, right? It's not a good habit to consume it, but how many of you have tried eating chewing gum? I'm sure all of you have tried, even if you say no over here. How many of you have tried eating chewing gum or what we say? When I was a kid, right, we used to have Boomer, bubble gum. Yeah, something of those names. I remember we used to chew and then we were making that bubble, right? Yes, then of course we will be chewing it and then we will try to make those balls small. Yes, oh, how many of you have tried it? Chingum. Yes. Not me. I can see a few of them are saying, ma'am, yes. Right? So similarly, if we look at that, right, so what happens in the Golgi apparatus, right? So when we talk about this, right, these structures are there. So of course the proteins and lipids are there. They will be moving from one stack to another. And what will happen once they are done, right? You know what will happen? They will come out in a form of balls like this. What we call these? We call them as the vesicle. Right? We call them as the vesicles. And what these vesicles have? They either have the proteins or the lipids. Clear? I'm not ignoring any one of you. Maybe I'm just too involved in the teaching, so maybe I might have missed your doubt. I'm really sorry. Please write your name. Write your doubt over here. People, ma'am, मैं तो बहुत ज़्यादा खाती हूँ, मुझे बहुत पसंद है. Okay. So everyone, the other part is over here, vesicle. Now let's understand the function of the ER. So the function of the Golgi apparatus, and everyone, I'm sure all of you have heard this. They are called as a postman. They are called as a postman of the cell. Yes or no? Right? They're called as a postman of the cell. And why they're called as postman of the cell? Because they are actually help in carrying materials. So what they do, they actually store, modify and pack materials in the vesicles and send to the different parts of the cell. So first of all, what they do, they receive the material from ER. So let's see everyone over here. Can you see? They will be receiving the materials, right? If I draw over here, wait a minute. So if we have ER, ER is making proteins and lipids. Okay? Now we have the Golgi apparatus. They will come over here. Now proteins and lipids, right, are in a very fancy way. And everyone, you tell me, for example, let's suppose you got an assignment and you're writing an assignment, right? But you know that you have it to explain it to a person who is in a small class, right? For that, what you will do, you will actually modify the topic, right? You will change the packaging, you will make the cutesy, you will make something really very cutesy thing on the top. You will modify the topic and then only you will give. Yes? So similarly, Golgi is very smart. He or she, I don't know what it we call all the organelles as it, right? So the Golgi apparatus, it's really very smart and super, super smart. What happens? Golgi is like, I know these all cell organelles are not that intelligent as me. I have to help them. I am the intelligent organelle. So you know what I will do? I will actually make the modification in proteins and lipids. So these chotu pavam organelles can understand and can do their function. Yes? So it's a very easy thing, right? So Golgi apparatus is making sure, right, that the message which is moving from ER, right, can be taken easily, right, and modify according to the needs of the cell. Clear? So from here it will go to the ER and then over here it will get modified. Let's suppose we're making it beautiful, right? This is our protein, right, and this is our ER. Sorry, this lipids. Now they have modified it. See over here the modified, and now they'll go to the cell. Everyone clear? That's a main function, right? That's a main function of the Golgi apparatus. Clear? Yes. Okay, so everyone remember this. Now, another function of the Golgi apparatus is that actually help in the synthesis of the lysosomes. Now, lysosomes, of course, are a very important organelle. We will discuss about them, but they are very, very important organelle. So, Golgi apparatus has two functions. Okay, everyone, who have any doubt, please just hold your doubt and let me revise this again. Everyone, focus over here. Do I have the focus of all of you? Yes? Yes, everyone. So, what we have Golgi bodies, right? So, Golgi bodies are very closely attached to the ER. Okay? They are the stack-like structure, parallel stacks, right? They have the space in between. I think someone was asking me what is lumen. These empty space in between, right? That is the lumen. Then of course they are stacked in parallel. What we call them as? We call them as cistern. Okay, we call them as cistern. Now what is the main function of the Golgi apparatus? It actually help in package, help in storage, and help in the modifying. Clear? And of course then the transportation, transport. So these are the important function of the ER. Apart from that, it actually help in the synthesis of lysosomes. Clear? Those of you who have doubt, I hope that this is clear. Okay, I can't spell you. Come on, what? Buddh. Lumen is nothing but the space, that the empty space that we have in the stacks that are present in the cisterns, right? That is called as lumen in the Golgi apparatus. Yes, Buddh. Yes, Buddh, I have answered your question. Arey, there no confusion. See, ER synthesize the proteins and lipids. Very huge structure is the ER. And Golgi apparatus is the best friend of ER, which actually help in making its function and taking all the material and sending it to different parts. Yes, Siachin. Sinchan. Okay. Sorry, Siachin I'm reading. Look at me, what is happening? Who will write their name as Siachin? But yeah, I don't know what I was thinking about a place. Yes, complex. Yeah, very good, Naman. Complex sugar. Yeah, that's a very interesting. Oh, hi Yash, how are you? After a long time. I know it's Shinchan, yeah. I can see a lot of news. Yes, a lot of new students. No, Dilip, it's not a membrane, bache. It's the stack. For example, if 1, 2, 3, 4, if we stack onto each other, we call it as a cistern. Yes, I know, it's a Sinchan, yes. Good to know that. Okay, everyone, let's move ahead. Now it's time for us to move ahead, everyone. Okay, let's talk about the next organelle. Everyone is my favorite. Ta-da! Say, girl, I'm not ignoring you. Yes, we're just moving to the lysosomes. Now, everyone, lysosomes are my favorite organelle because it's very easy for us to remember, right? Now lysosomes, if you look over here, right, are synthesized by Golgi apparatus, and the main function, right, main function of the lysosomes, everyone, is to make sure to keep the cell clean, right? To make sure there's nothing unwanted things that are there inside the cell. And we also call them as the suicidal bags of the cell. One of the most important function that we have to remember about the lysosomes is that they're the suicide bags of the cell, and let's understand why. So we know that lysosomes they are synthesized by the Golgi apparatus. Interestingly, everyone, they are membrane bound structure. That means that they just have single membrane. Everyone clear? CER. Golgi apparatus have double membrane. Right? We can clearly see in the structure that they have double membrane, but the lysosomes have a single membrane. So everyone, when we have a single membrane, how we write it? Membrane bound. Sac-like structure. Clear? Good. Okay. Now everyone, if we talk about what they do, they actually contains digestive enzymes, right? They have the digestive enzymes, and they actually make sure to digest the unwanted things that they have entered into the cell. Yes, so whatever is, for example, if there's a microorganism or if there's a waste inside the cell, lysosomes will take care of it. So they have a very important role. They have digestive enzyme, they actually help in clearing out the waste. Apart from that, we call them as suicide bags of the cell. And why we call them as suicide bags of the cell? Because just in case, if the cell is not functioning correctly, right? If the cell is not functioning correctly, if there's something wrong with the cell, what will happen? You know what will happen? Lysosomes will release all the enzymes that it has, and eventually what will happen? The cell will die. Cell will say, okay, bye-bye, I'm not functioning well. It's time for me to say bye-bye. That's why they're called as the suicidal bags. Clear, everyone? RER, rough endoplasmic reticulum. Very good, Lakshmi. Okay, everyone stay focused. Yes, RIP, rest in peace. Now let's quickly recall the functions of the lysosomes. Over here, everyone, remember, any foreign material entering into the cell, such as bacteria or anyone, will be taken care by the lysosomes. And of course, it makes sure that if there's any disturbance in the metabolism, if something is wrong with the cell, it will be releasing the enzymes, and eventually we'll see the death of the cell. Yeah, it simply clean the cell. Yes, absolutely correct. Okay, yes, Ramesh, you'll be joining the Telegram. Bye. Bye, Yash. Do take care of yourself. Sac is nothing but, basically this is a sac. A sac-like structure. Okay, this is small, like a small structure. Ma'am, how do lysosomes know that what to throw outside? They're very smart. So so when we talk about the cells, everyone, if you remember in the beginning, right, we discussed two important things. Intake and outtake, right? Phagocytosis. You remember what is phagocytosis? Right? Exocytosis and endocytosis. Yes or no? Cytosis. Yes, endo and exo. Exo means exit. Endo means entry. Endocytosis, engulfing the cell. Something is not engulfing the cell, engulfing or allowing the material to come inside. Exocytosis, moving out. Okay? Yes? Sac is bori. Yes. Yeah. Clear? So endocytosis, if a cell, right, the membrane will go out and will eat something, we call it as endocytosis. Taking inside the cell. Endocytosis. Exocytosis means throwing out. Clear? Good. Ankit, आप late आयो class में क्या? RER is rough endoplasmic reticulum. Okay. Everyone, are we clear up to here? Quick thumbs up. Thumbs up, thumbs up. Lysosomes have so many nicknames, yeah. Yes, clear. I can see thumbs up. So let's move ahead, everyone, to the next organelle. Okay, now you tell me. I'll play a riddle with you. I am an organelle that stores things. I am a huge in plant cells and small in animal cells. Who am I? Welcome to the class, Manpreet, Riddhi. Where do the enzymes come from? From rough ER. They are made up of proteins. They are coming from the rough ER. Vacuole. I think you all love vacuole, and here we have the answer. So everyone, the next organelle that we are discussing is the vacuole. Now what is a vacuole? Of course, it's a very important organelle that plays a very important role in the storage. It contains amino acid. Let's suppose I'm just writing over here, right? Amino acids, sugar, various organic acids. So right, so it is a place where we'll see the storage happening, right? Of course, in the plant cells, it's a very huge structure, right? But in animal cells, of course, it's not that huge. So it has a storage sac which store the solid and the liquid components. In the animal cells, we have more number of vacuoles, but the size of the vacuoles is small. In plant cells, there will be the one vacuole that will be occupying the majority of the space, right? Clear? Yes. This is a very important difference between plant cell and the animal cell. The last one, everyone, right? The central vacuole that we have in the plant cells, right, occupies up to 50 to 90% of the volume. And when we talk about the functions, apart from the storage, right, apart from the storage of the various different components, right, it plays a very important role in providing the turgidity and rigidity to the cell, right? It's because they are filled, right, with the liquid and with the solids. It actually helps in maintaining the shape of the plant cell. Everyone, are we clear? Yes, all of you. Two important functions of the vacuole. Help in the storage. Second, help in maintaining the rigidity and the turgidity of the cell. Awesome. Deepika, abhi bachche bola function. Yes, two functions. Everyone take a screenshot. These are functions. Awesome. Now many of you are asking them how. So of course, let's suppose everyone, I'm sure you would have seen a plastic bottle, right? Yes, I'm sure you have seen a plastic bottle, yes or no? How the plastic bottle is? Yeah, usually like this, right? Chotu sa plastic bottle. I don't know what is this. My drawing is definitely not good. So I'm sure you would have seen the plastic bottles which have the water. Right now, do you think that you can crush them when the water is inside? Yes, can you completely crush them? Not right. We will be able to crush, we will be able to move a little bit, right? But not completely. So similarly, when we talk about a vacuole, right? It has solid and of course it has fluid. So because of that, when there's any pressure on the plant cell, this plays a very important role in putting the pressure back. If the pressure is coming from here, it will be trying its best to push it down like this. Okay? So that's how it actually provides the rigidity and turgidity. Very good. Rigidity means, what is the meaning of rigidity and turgidity? Jaldi se bataiye. Very good, very good. I can see there are a few of you who are spamming everyone. Please pay attention to your class. There's no point of wasting your time. Everyone, what is the meaning of turgidity and rigidity? See, if you look at the words only, you will find the answer. Yes, very good, very good. Rigid means strong, right? Very rigid. Oh, she's very, when we talk about, oh, she have made a very rigid house. Very strong, a very hard house, right? Turgidity, on the other hand, when we talk in terms of the cell, means that when the cell will take more water, it will become swell, right? It'll change its shape. A little bit will have it, right? And that is called as the turgidity. Clear? Swollen. Very good, Gungun. Simone! Very good. Welcome to the class, but please don't spam. It doesn't get compressed easily. Absolutely correct, Naman. Very good, Siddhi. Shubha, it's okay. Gungun, yes. Very good, Lakshmi, Shraddha, Govind, Aman, Siddhi, Palak, Rabindra, Manpreet, Anshika, Ashi, Dwarka, Manpreet. Is it? Just change the setting of your phone. Yes, Shruti. Okay, very good. Yes. Budh, very good. Okay, so we are clear with this, right? Now let's move ahead, everyone. We have final 5 minutes. The final 5 minutes, everyone, and let's quickly take a look over here. Now, I'm sure all of you have seen this amazing yummy fruits. Yes or no? Yes or no? I'm sure you would have seen this, right? Yes, everyone? Plastics. Are we talking about plastic? And you are saying yes. You got the hint and you are talking about the organelle. Yes, very good everyone. So what we have over here are the different types of fruits, and of course they all are in a very beautiful, vibrant color. So what is the meaning of these? Like, of course, they have the very specialized pigments, right? Organelles, that they have pigments. Because of those, we have the color. So everyone, this is very important. So we call them as the plastids, right? We call them as the plastids. And we have 3 plastids in total. Everyone take a screenshot and we'll learn about it. Quickly take a screenshot, everyone. So plastids are of 3 different types that we will be studying in our class 9th. Okay? Now we are not going into that detail. You will be learning about these in your higher classes. So, but as of now, let's talk about. So we have 3 different types of plastid. Chloroplast is a plastid which has a green pigment. And of course, it actually helps in the performance or the process of photosynthesis, right? We will see that the leaves that are green in color have the pigment which is chlorophyll, right? And we have chloroplast in the leaves. So it's an important function, actually help in the process of photosynthesis. What is chromoplast? Now chromoplasts are usually the bright, vibrant pigment that we, they have the pigment which is the keratin, right, which gives the beautiful colors to flowers and to the fruits. Last one is a leucoplast, right? Now it has no pigment. Leucoplast does not have any pigment. That is why they actually help in the storage of starch, oil and lipids. They're usually found in the roots. Now everyone, tell me, have you seen any root, right, which is colorful? Usually, if you look at the roots, right, they are not that colorful. Yes, right? Yeah, I know white is a color, but yeah, we usually call them as colorless. Good. Okay. So everyone, are we clear? There are 3 different types of plastids. We have chromoplast, chloroplast and leucoplast. In our textbook, we have in detail about the chloroplast, right? And there's a very common mistake that we have in our mind, that the plastids are only in the cells of leaves, fruits and flowers. It's not that, right? They are actually present in all the cells of plants. We usually assume that a green color leaf will not have leucoplast or will not have this. But it's not the case, right? So plastids are present in all the cells of the plant, right? And of course, the number of these plastids may vary in each of the different parts of the plants. Okay, for example, the green leaf have greater number of the chloroplast and less number of leucoplast and the chromoplast. Everyone, are we clear? Yes? Good. Very, very good, everyone. I will take your doubts. Give me some time, right? Give me some time, everyone. Okay. Now, everyone, let's take a detailed look at the structure of the chloroplast. Everyone, we are talking about the chloroplast and we call it as the kitchen of the cell. Remember that. Cell sap. Cell sap is nothing but the fluid which is present inside the cell. We call it as a cell sap. Okay? Right? The fluid that we have in the vacuole, I would say, we call it as a cell sap. Fluid present inside the vacuole, the central vacuole, is called as a cell sap. Very good. Okay. So now we are talking about the structure of the chloroplast, everyone. So here we have the structure of the chloroplast. Right? The important thing over here is that they have double membrane. They have double membrane, very, very important. Then of course they have disc-like structures. Everyone, can you see this? Good, very good. See over here, the green color, we can see this disc, right? So this particular single disc is called as the thylakoid. What we call it as, everyone? Thylakoid. The single disc-like structure is called as thylakoid. And when we stack these thylakoids onto each other, we have a structure called as grana. Clear? Clear? So if I say this is thylakoid, right? If we say this is thylakoid, the stack of thylakoid all together is called as grana. Clear? Okay. Now we have 2 important things. Many of you are asking about it in the chat. Then ma'am, what is stroma? Now stroma is nothing but the fluid which is present inside the chloroplast. So over here, whatever the fluid is, that we usually call it as the stroma. Okay? And they also have the genetic material. So just like mitochondria have its own DNA, plastids also have its own DNA. So chloroplast have its own DNA. Everyone, are we clear? Yes. Nisha, more numbers, right? We have, see, one stack is called as granum, right? One stack is the grana, and of course, if we have more, we'll call them as. Very good, awesome. Yes, okay. Now let's move ahead, everyone, to the next part, right, which is related to the chloroplast only. We know that chloroplast is only present in the plant cells and not in the animal cell. Clear? Everyone clear? Yes? Where we have the chloroplast? Yeah, I know time's up. Give me 1 more minute. Right? Plastids are present in the plant cells, right? They have 3 different types that we are studying in class 9th. We have chloroplast, a chlorophyll pigment which gives green color, helps in photosynthesis. Chromoplast have the pigment keratin which gives a very beautiful and bright color. Leucoplast has no pigment, that's why no color, but it actually helps in the storage of starch oil. Good. The last one, everyone, this is a bonus thing for you. Ta-da! In animal cells, we don't have plastids. I don't know why you're asking, ma'am, do we have plastids in the animal cells? No, we don't have plastids in animal cell. Okay, same function, the function of vacuole in animal cells, storage. Okay. Good, everyone. So now, everyone, the last one over here is the centriole. It's not mentioned in our textbook, it's an extra information for all of us because you'll be learning this in a small bit in the cell division. So centrioles are a very special organelles, right? They have this structure, you can see. Basically, in a structure, in a one centriole, we have 2 structures, right over here, 1 and 2. They occur in pair and they actually help in the formation of spindle fibers. These yellow color thread are called as spindle fibers and they help in the cell division, okay? So just letting you know, everyone, you can read more about it, but this is very interesting. Clear? Yes, very good. Yes, very good, very good. Okay, everyone, are we clear? Right, so let me quickly revise this for you. All of you just pay attention now. Yes, I will show you the timetable. Shubha, I will show you the timetable. Okay, everyone, so we started, right? So we started, everyone, with the ER. Okay? Okay, everyone, I'm just making a very rough mind map. Please stay with me. Okay, you help me jaldi say, everyone. What is ER? ER is endoplasmic reticulum. It is of 2 types, smooth ER and rough ER. Smooth ER produce or manufacture the protein and rough ER produce the lipids, right? These 2 together help in the formation of membrane and we call it as the membrane biogenesis. Lipids, right over here, SER, help in the detoxification also. Clear? Very good. Now then we talked about the Golgi apparatus, right? So ER, say information will come to the Golgi apparatus, right? They are the stack-like structures which we call them as the cistern, right? They actually help in they pack, store, modify and transport. Yes, clearly everyone? Golgi apparatus help in the synthesis of the lysosomes and lysosomes are called as the bags of the cell. What they have? They have the enzymes. Which enzyme? Digestive enzymes. Clear? Clear, everyone? Are we clear with this? Good, everyone. Yes. Now, I'm just rubbing it off. Okay, I'm just rubbing it off. You can go back and see this. Then let's talk about mitochondria. Beautiful structure, right? They have these folds, and what are these folds called as, everyone? Cistae, right? The folds are there to increase the surface area, right? And what they do, ATP synthesis. Clear? Yes, very good. We call them as the powerhouse of the cell. Have own DNA. Ta-da! Okay. Take a screenshot, everyone. You can come back also. Now I'll rub it off and let's talk about plastid. I can see few of you were requesting me to repeat it again. So here we are, everyone. What are plastids? Plastids are there in the plant cells. Only in plant cells. 3 different types. Can you quickly tell me, everyone, what are the 3 different types? We have chloroplast, pigment, right? Gives green color, helps in photosynthesis. Then we have chromoplast. Pigment is keratin, right? Colored parts mein we will find this. Fruits and flowers. Then last one, everyone, leucoplast. No pigment, right? And of course, we will see it actually help in the storage of oil, starch. Clear? Okay. Then of course, everyone take a screenshot of this. Vacuole, storage sac. Main function and help in providing the rigidity and the turgidity. Okay. And then of course we studied about the centriole which help in the cell division. Suhani enzymes are nothing but they are made from the proteins and we call them as the catalyst. So in any reaction when they come they increase the rate of the reaction. Yes, easy peasy. Everyone, with this we are done. Now I want you to write the answer for this question. This is very tricky. You have to do a lot of research. Which organelles have single membrane structure and which organelles are double membrane? So please do write answer in the comment section below. I'll be waiting for this answer. This is very tricky. So all of you have to give the correct answer. Do a little bit more research because this is a little bit confusing. So I want to see whether you are doing your research or not. Okay, everyone? So, right? Easy peasy now. I can see you're saying easy. No, of course, we have other sessions for this particular chapter. Yes, good. Don't worry. Yes, Asta, that was a doubt board. Please ask your doubt. I'm sorry. Didn't have a separate doubt board, but yeah, I was just taking your doubts. Bimal, what do you think? Yes. Ma'am, if the natural colors are made from flower, then... What? It was an interesting question. Asta, ma'am, if the natural colors are made from flowers, it means that colors have plastids. Oh, interesting question, right? It has the pigment for sure, right? So we have some connection over there. Both spindle fibers are the structures, right? The thread-like structure that are released by the centriole, made by the centriole during the cell division. You will learn more about them in your higher classes. Okay, everyone. Yes, I'll show you the timetable. So everyone, with this, I hope that everyone you have enjoyed the class, right? Everyone, please make sure to hit the like button for the video and do share it with your friends. If you're new here, do take a moment and subscribe to the channel. Right? I can see that many of you are still have doubt. Everyone, I'm really sorry. I have a class at 7 p.m. class 10th on this same channel. So I have to leave now. It will be great if you write the doubts in the comment section below. I'll read them and will address them or I'll reply there. Okay? Yes? Thank you so much, everyone, for understanding. I hope that you've enjoyed the session, everyone. Do take care of yourself, right? And I will be waiting for your doubts. Don't worry, I'm sorry if I've missed your doubts. I will be waiting for your doubts. Please write it over there, I'll answer. And we'll meet really very soon. So do take care of yourself, everyone. And keep on learning with BYJU'S. Bye-bye.